Welcome back. Today, let's go over the histology of the testes. So remember, the genitalia of both males and females have an internal and external component, and the internal component of the males would include the ducts, the accessory sweat glands, and the testes, which seems like the testes would belong to the external genital organs because it lies outside of the the abdominal cavity, of course. But it's considered internal because there isn't a direct outlet to the outside of the body. As you can see here, the testes lie within the scrotum, and then it has to go through the abdominal cavity, through the prostate, and then it can leave the urethra. So there are the genital ducts, which are the intratesticular straight tubules that leads the sperm outside of the testes and into the excretory ducts, which is like the epididymis, and then goes into the vas deferens, and then the ejaculatory ducts, and then the urethra itself. The accessory sweat glands will secrete, lubricating fluid. And then the only external organ is going to be the penis because it contains like the urethra. Okay, so development-wise, it comes from this coelomic epithelium and the mesenchyme. So there's two components. That's why the male sympathetic system, not just sympathetic system, I'm sorry, the innervation is both sympathetic and parasympathetic. We'll go over that later, but it's due to the two different embryologic origins. So the primordial germ cells are going to come from the epibast, and then that induces the cells of the coelomic epithelium and the Seminiferous to make the primitive sex cord. So it comes from the epithelium and some of the urinary tract, like primordial tissue. So the sex cords are going to be pre-Sertoli cells, so they're not quite matured into Sertoli cells, and they secrete the anti-Mullerian hormones. And of course, without the Mullerian hormones, the Mullerian ducts will degenerate, and then the Sertoli cells will be mature, and then the mesenchymal cells will be differentiated into the Leydig cells. And then the Sertoli cells are going to be influenced through a testosterone surge, like a mini one, during development, and that forms the testes cords. And then during puberty, it's actually going to canalize to make the seminiferous tubules. Oh, sorry. So before the canalization, the children cannot, not just produce sperm, but they can't ejaculate itself because the canalization hasn't happened. Okay, so there's two different functions: the exocrine function, which is the spermatogenesis, so making the sperm, and the endocrine function is going to be the testosterone secretion. So testes itself is made out of many, many seminiferous tubules, and all of that is going to be contained and supported by a connective tissue stroma. And then the stroma is going to be continuous with the tunica vasculosa testes. So there is going to be the vessels associated with it. And then on the outside is the tunica albuginea, which is the capsule of dense connective tissue, and it surrounds the each testes. And those are going to be thicker towards the bottom, and that makes the mediastinum testes. Okay, so the mediastinum testes is the thicker part and is covered by the tunica vaginalis, which is a mesothelium, and that's supported by the fibrous connective tissue. And those are permeated by the ret testes, and that's where the vessels are, which can be bad because during inflammatory processes, this is where it is going to be swollen and inflamed. It's because of the location of the vessels. And then the mediastinum itself is going to be lined on the inside by an endothelium, and then they send in these septa that separate the testes into lobules. And then in each lobules, it has the seminiferous tubules that are surrounded by connective tissues and Leydig cells. So the seminiferous tubules are going to they're going to induce formation of or maturation of the sperm and whatnot, thanks to the hormones of the Leydig cells. So the interstitial tissue is going to be like reticular fibers, and it contains quite a bit of things. And that so it's going to contain vessels for nutrition and waste removal, along with cells that support the tissue itself, and immunological cells. The Leydig cells are going to be cuboidal and round, and they have a lot of fat droplets. So those are going to secrete steroid hormones, of course, and it's going to be acidophilic and have a lot of mitochondria, and it's going to be stimulated to make the testosterone from the chorionic gonadotropin, so that comes from the mom. And then over time, they get yellow because of the accumulation of the lipofuscin droplets. The seminiferous tubules are going to continue into the straight tubules, and then from there it goes into the red testes and the epididymis, and there's many of them to convey sperm, and that's just their job. They just kind of contain the sperm for a short period of time and allow the passage into the epididymis, where the sperm is stored for a longer period of time. So the walls of the testes is going to be made out of tunica propria. That's the outermost. That's going to be the outermost layer, and it has only a few fibroblasts, and they really work as boundaries because the cells are sort of muscular, but they form a robust layer. In between the myoid cells are going to be the actual germ cells, and the germ cells are going to be the ones that differentiate into the spermatozoa, and then the spermatozoa are pushed out once matured to a certain extent. The epithelium of the testes is pretty complex, and it's made out of a specialized stratified epithelium, which is a germinal or spermatogenic epithelium. So the spermatogenic epithelium will support the sperm cells and push them out once matured enough. Those will contain the spermatogenic, which are the precursor to sperm cells, and the Sertoli cells, which support those. Sertoli cells are your permanent cells, and they don't mature or die. They're present before the birth, and they remain until your death. And they have very interesting cytoplasmic processes, and they make these deep invaginations, and those will hold your spermatogenic cells or your germ cells. They extend quite deep from the basal lamina to the lumen, and they're contained or they're made out of these columnar cells, and there's going to be there's a oval nucleus, and the chromatin is going to be euchromatin, so it's going to be somewhat condensed. And there's a prominent nucleolus and a lot of other organelles. So the Sertoli cells are just right around here. So the testes form a protective barrier, whether from the outside or internal environment, and more importantly, it protects from our own bodies. They form the two different compartments, a basal compartment, which it will have their Spermatogonia because they have a proper chromosomal amount, and then the other compartment, the adluminal compartment, have the germ cells that don't have the normal chromosomal count, and that is amount, and that is completely sealed off from the body because the body will attack these cells, leading to a sort of autoimmune. I say autoimmune because it's your own body attacking your own germ cells, but these cells are not your own, if that makes sense. These cells are going to be your children's cells. But anyway, if the blood testes barrier fails, it's going to lead to infertility because the immunologic attack is going to just wipe out all of the germ cells. So back to the basal compartment, it's going to have the primary spermatocytes disassembling the adhesion molecules of the occluding junction, and then that kind of like moves in while still adhering to the Sertoli cells. So like they're still connected, but once matured enough, it goes from the basal compartment to the adluminal compartment. So of course, the cytoplasmic enfoldings like literally cup the cells, and there's going to be nutrition and metabolites to be exchanged. There are the inhibins that are created that decrease the FSH production and release, and in order for the right amount of sperm cells to be created and for them to mature. And then there's of course phagocytosis to get rid of residual bodies and defective cells. So your spermatogenic cells come from progenitor cells, which are your Spermatogonia. And spermatogenesis in males occur from puberty until death, and it's all through mitosis, and those will make the diploid spermatogonia, and then meiosis will make the 4 haploid spermatids per spermatocytes. So they make up quite a lot of cells because it's all multiplied by 4. The spermatogenic cells make up a lot the concentric layers, so they kind of build on each other in the seminiferous epithelium, and then they're all connected to each other in a cytoplasmic bridge. We're not really sure what the cytoplasmic bridge is. Some people think it's for communication, some people think it's for the exchange of nutrients, but it exists and they're all connected. So here are your Sertoli cells. They're a little bit larger, and here are your Spermatogonia with the condensed nuclei. Here are your primary spermatocytes, which it's getting a little bit more diffuse. Spermatids are your smaller cells. Okay, so Spermatogonia, it's a 2N, so it is diploid, and it's going to be your cells in the seminiferous tubules before puberty. So there's 2 types. Type A is going to lie in the basal compartment, and they divide by mitosis in order to be either type A or type B cells. So there's going to be asymmetric. There's going to be some asymmetric division, and then the type B cells are more mature. They're lying next to the basement membrane, and they differentiate into the primary spermatocytes. So as you can see, this is how you make more and more and more. So the type A cells aren't going to be they're not going to be reacted to because they have the same chromosomal number as somatic cells. So that's important to know. Primary spermatocytes are diploid, and they are going to have 4N, and they have abundant cytoplasm, a large heterochromatic nucleus, as you can see, and these are the largest of the spermatogenic cells, and they're found in the tubular epithelium. The secondary spermatocytes are going to be your haploid cells, and those are going to be found also in the epithelium, and they don't really last long. They kind of go under the second meiotic division almost immediately after they come up, so they don't arrest in the females. Spermatids are going to be haploid, and they only have 1N, and they are much smaller, as you can see here. The chromatin is going to be granular, and they condense even more, and they're found more in the lumen of the tubule, and then they become the spermatozoa. So there are a lot of structural changes, and those aren't cell divisions. This is just structural changes, and that includes the formation of the acrosomes, and the mitochondria move down towards the flagellum, and there's continuous condensation of the chromatin into the head, and then a lot of the cytoplasm is shed and then is reabsorbed in part of the epididymis. So that's the excess fluid that it reabsorbs. Okay, finally, down to the spermatozoa. Those are going to be haploid, and these are motile now, and they are there are two different types. The testicular spermatozoa aren't quite mature yet. They can't swim, and they can't be capacitated yet. Once they are matured, they can swim and they can capacitate, and that usually occurs in both male and female genital tracts. So like the sperm can mature still swimming through the urethra during their ejaculation, and in the vagina when it's deposited. Okay, so this is the acrosome reaction. There's going to be some organelle containing the hyaluronidase and the acrosome. It forms during the spermiogenesis in the cytoplasm, and then it's going to like form the membrane over the nucleus and the plasma membrane, and there's a neck that's also formed. And the midpiece and the tail will contain the most of the mitochondria. Okay, so the ducts are going to be lined by simple cuboidal epithelium, and there aren't any spermatogenic cells, of course. There's only Sertoli cells, and they are going to change once the ducts are in the ret testes. Now they are low cuboidal, and those will open into the efferent ducts, and in those they're going to be ciliated, and there are some non-ciliated cuboidal cells as well, and that makes the wavy outline of the lumen. The non-ciliated cells absorb the excess fluid, and then the ciliated cells will like propel the immature spermatozoa into the epididymis, where it can mature. So that's it for now. Thank you for your attention, and good luck studying.